Alright guys, this video is going to finish up our cell membrane and transport notes. And so last time when we covered all of that information, we focused on passive transport. And passive transport is when you are moving substances from a high concentration to a low concentration. It does not require energy because they are moving in a natural way, which is kind of downhill or with that concentration gradient. And so this particular portion of the notes is going to focus on the other main type of transport, which is going to be active transport. And so it's going to be just the opposite of your passive. Where passive was high to low, active is going to be low to high. Where passive did not require energy, active transport does require energy. And this is pretty easy to remember because we need energy to be active. And so anytime we are moving molecules where they naturally do not want to go, we can call this against the concentration gradient, or we can call it up the concentration gradient. If you want to, you can think about it like a river. The river naturally flows in a particular direction. That is passive. No energy has to be put in for the river to flow. But anytime you want to go upstream, you're going to have to put in some energy to go that direction. And so that's essentially what's happening in active transport. We're moving molecules from where they are lower to where they are higher, and that is not a natural movement. And so this is going to require energy. Now, we did have some proteins that were involved in passive transport. You could have carrier proteins, you could have channel proteins. But here we're also going to see proteins involved, but in this case, we typically call them pumps. And so the word pump, anytime you see that, it automatically implies active because you need energy to pump something, okay? In a carrier protein or a channel protein, a lot of times there's just a flow, whereas with the pump, we're actively moving them against that concentration gradient. And so, as always, the proteins that are doing this are very specific. Proteins always have a specific shape and do a very specific job. And so it's not like there's just a general pump. There's going to be a specific protein pump that will move specific molecules. And the energy that we need is going to be supplied by ATP. So we looked at that back when we did metabolism, and we know that one of those phosphates is going to break off, and it's going to phosphorylate the protein, which means energize the protein. And so that's when that phosphate is transferred. That's called phosphorylation. And that's what's going to allow that substance to be pumped across the membrane. So one common example of a pump in our cells is sodium-potassium pump. It's actually really important when it comes to electrical impulses and nerve impulses, action potentials. It's a pretty important pump that we have in our cells, especially our neurons. And so what that sodium-potassium pump does is it always pumps 3 sodiums out and 2 potassiums in. Right now you really don't have to know why or understand the purpose of it, but it actually has a very significant purpose, and that's maintaining resting membrane potential in a neuron or nerve cell. So all you need to know at this point is that when you see pump, you know that it is active transport. And in this case, we are specifically moving sodium and potassium. And so here's an example of what it would look like. So in order for this pump to work, ATP has got to be introduced. And so that phosphate is transferred to that protein, making ATP into ADP. And anytime you're looking at a picture of active transport, you're most likely going to see ATP in the picture. And so as you can see that energy is applied, we have 3 sodiums that are moving out, and we have 2 potassiums that are moving in. Now, something else to understand a little bit here, and we'll explain this more when we get to cellular respiration, but all membranes have a voltage, okay? There is a difference in charge across the membrane. So just like there can be a concentration gradient, where there's a difference in concentration, there can also be a difference in voltage or charge, and that's what we call an electric gradient. And so just like with concentration, molecules like to move a particular direction based on concentration. Molecules will also like to move in a particular direction based on charge. And so in general, the cytoplasm is more negative than the outside of the cell. And so we have this charge difference. And so the combination of our concentration gradient along with that electrical gradient, and we have what's called the electrochemical gradient. And so an ion, or those charged particles, may diffuse down that gradient, just like they would diffuse down an electrical gradient. Now, we often see these charges come into play with these electrogenic pumps. And like I said, we're going to explain this further when we get to cell respiration. So the sodium-potassium pump is one. We have 2 positive ions going out for every 2 positive ions that are going in. We also have another example called a proton pump. And a proton is essentially a hydrogen ion. A hydrogen only has 1 proton and 1 electron, so when it loses that electron, you pretty much just have a proton. And so what these electrogenic pumps do is they essentially store energy. And so if you think about it like that river again, that river has a natural flow, okay? There's energy and that kinetic energy. But if we were to dam the river and we were to stop that flow, then what happens is we are creating lots of potential energy. And so what electrogenic pumps do is they essentially create potential energy. They store energy that can be used to do work. And we're going to see this come into play when we're actually making energy for our cells. So this proton pump especially is going to be important in cellular respiration. Once again, for now, the main thing you need to know is that when you see pump, if you see that word, it is going to be a type of active transport. We are moving molecules against their concentration gradient. We are moving them from where they are lower to where they are higher, and that is going to require energy. But we will see some of these coming into play later on. Now, sometimes we'll actually have 2 substances moving together, and we call that co-transport. And so this is where we actually have the flow of one is going to power the other. So as I mentioned earlier with the river, there is energy in the flow, okay? So there is kinetic energy as molecules move down or as just any molecules move, period. So if we're talking about, active and passive transport, sometimes energy is going to be used, as you can see here in the picture. Energy is used to move those substances, okay? And so this is how we create that potential energy. So when we pump something to the other side of the membrane and we store it, it's kind of storing energy. And so what's happening here in the picture is you've got ATP being used in active transport to pump those protons across. Now, there's another protein here that is going to allow the downward flow, that passive transport of those protons back down. Well, because there's energy in the flow, as those protons flow down, that provides energy to pump another substance across. So as one substance flows downhill, that is going to power the uphill movement that requires energy. And so that's what co-transport is. As one molecule flows down its gradient, that's going to power energy to move another substance up the concentration gradient. So you still do have active transport involved here, but you have 2 molecules kind of moving together. Now, there's a couple other types of active transport, and these are a little bit different in that we are not moving across the membrane. These types of movement are going to be literally using the membrane to either bring things in. Notice we have endocytosis. This literally means into the cell. Okay, cyto means cell, endo means inside. So endocytosis is a process of bringing substances into the cell, and exocytosis is going to be the process of moving substances out of the cell. Now, the way these are different is because we're not moving across the membrane directly. Anything that we're moving here is going to be very large, and we're going to move it by what we call bulk transport. So we don't just have like small ions or substances moving across the membrane. We literally have the cell using the cell membrane to either surround and engulf. This word right here implies that it's endocytosis. The cell is surrounding and engulfing, or the cell is expelling particles via the cell membrane. And this does require energy, so this is a type of active transport. You may want to add that to your notes. Both of these are examples of active transport. Because when you see a picture of it, you're going to understand that it's going to take energy for the membrane to be able to do this. So endocytosis is bringing things into the cell. There's actually 2 types. We have phagocytosis and pinocytosis. Phago is bringing in food. Pino is bringing in liquid. So phagocytosis is a form of cell eating, We actually have cells in our body known as phagocytes. They are a type of white blood cell, and their job is to go around and eat pathogens. They're a type of white blood cell that help us in our immune system. And so what those phagocytes do is they kind of surround a pathogen or a bacteria with their membrane, and then they bring it into a little vesicle, and then they attach it to a food vacuole, and then they essentially just digest it. Pinocytosis is similar. You're going to have a little pocket forming in the membrane. It's going to fill up with liquid, and then it's going to pinch off and come in as a vesicle. So both of these are types of transport involve vesicles, which are just little membrane sacs. And so exocytosis will be the opposite, where endocytosis is bringing in either food or liquid. Exocytosis is going to be expelling them. And I just think pictures help a lot, so we're going to see a picture here in just a second. Now, another example is receptor-mediated endocytosis, and this is where it's just a more specific version. This is where before endocytosis can happen, a ligand or a signal molecule has to bind to a protein receptor. Otherwise, the cell will not engulf. So it's just a little bit more specific in that you have to have a particular signal that's going to kind of trigger that endocytosis process. It will be very specific, but the ligand does have to bind to the protein before endocytosis will happen. And so just to kind of show you some pictures, here you have a food particle being surrounded. And so in order to do this, you kind of have to have this movement, this pushing of cytoplasm against the membrane to create these little extensions. And that's where energy is going to come in, okay? So these are going to kind of surround, they'll kind of go all the way around. And then you can see that it's going to bring that food particle in a little vesicle. It'll take it and it will digest it. Pinocytosis, you have these little pockets forming. Once again, we have that membrane kind of enclosing, and then it brings it in. And then here we have receptor-mediated, where before it's going to happen, we do have to have these specific molecules that bind to the receptors, and that is what will trigger, but we still have this movement in. And then exocytosis is just going to be the exact opposite of that. And so that actually finishes up our cell membrane and transport notes. Hopefully we have a pretty good understanding. You are going to be getting your pre-lab today, and the next 2 days are going to be spent working on lab, looking at diffusion and osmosis across a membrane. I hope you guys are doing well, and I will probably be there to set up lab tomorrow, so hopefully I will see you then. Y'all have a great day.